Hello, everyone. Welcome to today's lecture on a topic that is profoundly impactful in medicine and a subject of immense global concern: antibiotics. We'll be delving into their fascinating mechanisms of action, how these remarkable compounds target and defeat bacteria, and, crucially, the growing challenge of antibiotic resistance. I'm thrilled to guide you through this journey into the microscopic battlefield of microbial warfare. Think about it. Before antibiotics, a simple cut could lead to a life-threatening infection, and common illnesses like pneumonia were often fatal. The discovery of antibiotics truly revolutionized medicine, dramatically extending human lifespans and transforming how we approach infectious diseases. They are, in many ways, one of humanity's greatest scientific achievements. However, this incredible success story also comes with a significant challenge: the rise of antibiotic resistance, often dubbed the superbug problem. This isn't just a scientific curiosity, it's a pressing public health crisis that demands our understanding and attention. Over the lecture, we'll first establish a foundational understanding of what antibiotics are and their historical significance. Then, we'll dive deep into the molecular and cellular strategies antibiotics employ to kill or inhibit bacteria. Following that, we'll shift our focus to the equally complex and concerning phenomenon of antibiotic resistance, how bacteria fight back, and why this is such a grave threat. Finally, we'll conclude by considering our collective role in preserving the effectiveness of these vital drugs for future generations. So, let's get started on understanding these magic bullets of modern medicine. Simply put, antibiotics are antimicrobial drugs specifically designed to treat infections caused by bacteria. Their primary function is either to kill bacteria outright, a property known as bactericidal action, or to inhibit their growth and reproduction, which is bacteriostatic action. This distinction is important because it influences how they're used clinically. Now, a crucial point often misunderstood by the general public: antibiotics are ineffective against viruses, fungi, or other non-bacterial pathogens. Taking an antibiotic for a common cold, which is typically viral, is not only useless but also contributes to the problem of antibiotic resistance, as we'll discuss later. This specificity is a cornerstone of their utility. The story of antibiotics is truly one of the most compelling in medical history. While ancient civilizations used natural remedies with antimicrobial properties, the modern era of antibiotics truly began with an accidental yet monumental discovery. In 1928, Alexander Fleming, a Scottish physician and microbiologist, made a serendipitous observation. He noticed that a mold, Penicillium notatum, contaminating one of his Staphylococcus bacterial cultures, seemed to inhibit the bacteria's growth around it. This humble observation led to the isolation of penicillin, the world's first true antibiotic. Fleming's discovery, while initially challenging to purify and mass-produce, marked a turning point. Its clinical application during World War II, largely due to the work of Howard Florey and Ernst Chain, transformed battlefield medicine and ushered in what's often referred to as the golden age of antibiotics. Suddenly, diseases that were once death sentences became treatable. This era saw the rapid discovery and deployment of numerous new antibiotic classes, fundamentally changing the landscape of infectious disease management and leading to incredible improvements in public health globally. So, what makes antibiotics so special, allowing them to combat bacterial infections effectively without causing undue harm to the patient? The answer lies in a fundamental principle known as selective toxicity. This concept is the cornerstone of effective antimicrobial therapy. Selective toxicity means that an antibiotic must preferentially target and harm the invading bacterial cells while causing minimal or no damage to the host's own cells, our human cells. It's like firing a magic bullet that specifically seeks out the enemy without collateral damage. How do they achieve this remarkable feat? The key is that antibiotics exploit structural or metabolic differences between bacterial cells and human cells. Bacteria are prokaryotic organisms, meaning they lack a nucleus and other membrane-bound organelles, and they possess unique structures and biochemical pathways that are absent or significantly different in eukaryotic human cells. Let's use an analogy. Imagine you have a very specific key that can only unlock one type of lock. In this analogy, the antibiotic is the key, and a specific bacterial component is the lock. Our human cells lack that particular lock, so the key, the antibiotic, cannot interact with them. For example, bacterial cells have a rigid cell wall composed of peptidoglycan, which is absolutely vital for their structural integrity and survival. Human cells, on the other hand, do not possess a cell wall. Therefore, an antibiotic that targets the bacterial cell wall can effectively kill bacteria without harming our cells. This is a perfect illustration of selective toxicity. Similarly, bacterial ribosomes, which are essential for protein synthesis, are structurally distinct from human ribosomes. This difference allows certain antibiotics to disrupt bacterial protein production without interfering with our own cellular machinery. Understanding selective toxicity is not just academic. It explains why these drugs are so valuable and highlights the ingenuity required in their design. It's the reason we can take these medications and expect them to cure an infection rather than causing widespread harm to our own bodies. This principle guides all antibiotic development and is crucial for their clinical utility. Now that we understand the principle of selective toxicity, let's dive into the fascinating how of antibiotics, their mechanisms of action or MOAs. While there are many different individual antibiotics, they can largely be grouped into a few major classes based on the specific bacterial processes or structures they target. Understanding these MOAs is fundamental to comprehending their effectiveness and, later, how bacteria develop resistance. Think of a bacterial cell as a miniature factory, constantly performing vital functions, building its walls, making new proteins, replicating its DNA, and maintaining its internal environment. Antibiotics are designed to disrupt these essential processes, effectively shutting down the factory. We'll focus on the four most common and significant categories of antibiotic mechanisms of action. Each category targets a different vital component or pathway within the bacterial cell, leveraging those structural or metabolic differences we just discussed. As you can see from the image, we have various critical components of a bacterial cell: the cell wall, the ribosomes responsible for protein synthesis, the DNA and RNA involved in genetic information flow, and the cell membrane that regulates what enters and exits the cell. Each of these represents a potential Achilles heel for bacteria, a point of vulnerability that antibiotics can exploit. The first three mechanisms we'll cover: inhibition of cell wall synthesis, inhibition of protein synthesis, and inhibition of nucleic acid synthesis, are typically bactericidal, meaning they directly kill the bacteria. The fourth, disruption of cell membrane function, can also be bactericidal depending on the specific drug and bacterial species. By the end of this section, you'll have a clear picture of the diverse ways these molecular warriors achieve their mission of eradicating bacterial threats, laying the groundwork for understanding the challenges posed by antibiotic resistance. Our first major mechanism of action, and arguably one of the most important and safest, involves the inhibition of bacterial cell wall synthesis. The peptidoglycan cell wall is an absolute marvel of bacterial engineering. It's a rigid, mesh-like structure that encases the bacterial cell membrane, providing crucial structural support and protection against osmotic lysis. Think of it like a pressure suit for the bacterium. Human cells, importantly, do not have a cell wall. This makes the bacterial cell wall an ideal target for selective toxicity. Antibiotics in this class work by preventing the bacteria from properly constructing or maintaining their peptidoglycan cell wall. Specifically, they often interfere with the enzymes responsible for forming the crosslinks in the peptidoglycan mesh. Without these crosslinks, the cell wall becomes weak, fragile, and permeable. What happens then? Bacteria typically live in environments that are hypotonic relative to their cytoplasm, meaning there's a higher concentration of solutes inside the cell. Water naturally rushes into the cell due to osmosis. A healthy cell wall can withstand this internal pressure. However, when the cell wall is compromised by these antibiotics, the internal pressure becomes too great and the cell simply lyses or bursts open, leading to bacterial death. This makes these antibiotics primarily bactericidal. The most famous and widely used examples in this class are the beta-lactam antibiotics, which include penicillins, like amoxicillin and penicillin G. They bind to and inactivate enzymes called penicillin-binding proteins, PBPs, which are transpeptidases essential for cross-linking peptidoglycan strands. Cephalosporins, a large and diverse class structurally similar to penicillins, also targeting PBPs. Examples include cephalexin and ceftriaxone. These drugs are incredibly effective and have a generally low toxicity to humans precisely because they target a structure unique to bacteria. Their widespread use, however, has also unfortunately led to significant resistance issues, which we'll discuss shortly. But at their core, they represent a brilliant strategy for defeating bacteria by attacking their fundamental structural integrity. Moving on, our second major mechanism of action involves targeting the bacterial machinery responsible for protein synthesis. Proteins are the workhorses of any cell, performing virtually every function, from building structures to catalyzing metabolic reactions. Without proteins, a cell cannot survive or reproduce. The target here is the bacterial ribosome. This is another fantastic example of selective toxicity. While both bacterial and human cells have ribosomes, they are structurally different. Bacterial ribosomes are classified as 70S ribosomes, composed of 30S and 50S subunits. Human ribosomes, on the other hand, are 80S ribosomes made of 40S and 60S subunits. This subtle but crucial difference allows antibiotics to selectively bind to and inhibit the bacterial 70S ribosome without significantly affecting our own 80S ribosomes. These antibiotics work by binding to either the 30S or 50S ribosomal subunit, thereby interfering with various steps of protein synthesis. This disruption can lead to a variety of effects, preventing the initiation of protein synthesis, blocking the elongation of the polypeptide chain, or causing misreading of the genetic code, leading to faulty proteins. Let's look at some key examples. Tetracyclines, e.g., doxycycline. These drugs bind reversibly to the 30S ribosomal subunit. Their action specifically blocks the attachment of transfer RNA, tRNA, to the A site of the ribosome. This prevents the addition of new amino acids to the growing polypeptide chain, effectively halting protein synthesis. Macrolides, e.g., erythromycin, azithromycin. These antibiotics bind to the 50S ribosomal subunit. They typically inhibit the translocation step, where the ribosome moves along the mRNA, preventing the elongation of the protein. They can also inhibit the formation of the peptide bond. While many antibiotics in this class are bacteriostatic, meaning they inhibit growth rather than directly killing the bacteria, they effectively stop the infection by preventing the bacteria from multiplying and performing essential functions, allowing the host's immune system to clear the remaining pathogens. This strategic interference with a fundamental cellular process highlights another elegant way antibiotics disarm bacterial invaders. Our third critical mechanism of action targets the very blueprint of life for bacteria, their nucleic acids, specifically DNA and RNA synthesis. Just like all living organisms, bacteria need to replicate their DNA accurately for cell division and transcribe DNA into RNA to produce proteins. Interfering with these processes is a highly effective way to halt bacterial growth and survival. Antibiotics in this class work by inhibiting key enzymes involved in DNA replication or RNA transcription. By disrupting these fundamental processes, they prevent the bacteria from multiplying or from producing the necessary proteins and other molecules required for their survival and function. Let's explore the main examples. Fluoroquinolones, e.g., ciprofloxacin, levofloxacin. These are potent antibiotics that primarily target bacterial DNA gyrase and topoisomerase 4. These enzymes are crucial for DNA replication, repair, and transcription. DNA gyrase, for instance, is responsible for unwinding and supercoiling bacterial DNA, which is essential for it to fit within the cell and for replication to proceed smoothly. By inhibiting these enzymes, fluoroquinolones cause breaks in the bacterial DNA and prevent its proper replication, leading to cell death. This makes them largely bactericidal. Rifampin, or rifampicin. This antibiotic is particularly important in the treatment of tuberculosis. Its mechanism of action involves inhibiting bacterial DNA-dependent RNA polymerase. This enzyme is responsible for synthesizing RNA from a DNA template, the process of transcription. By blocking RNA polymerase, rifampin prevents the bacteria from producing messenger RNA, mRNA, which is needed to make proteins, and ribosomal RNA, rRNA, which forms ribosomes. Without these essential RNA molecules, the bacterial cell cannot synthesize proteins and quickly dies. Rifampin is also primarily bactericidal. These antibiotics are vital because they target pathways that are distinct enough in bacteria compared to humans to offer selective toxicity, although some can have more significant side effects than cell wall inhibitors due to the closer structural resemblance of DNA and RNA polymerases across domains. Nevertheless, their ability to directly interfere with the core genetic machinery makes them incredibly powerful tools in our arsenal against bacterial infections. Finally, our fourth primary mechanism of action involves the disruption of the bacterial cell membrane. While the cell wall provides external rigidity, the cell membrane is the crucial barrier that encloses the cytoplasm, regulating the passage of nutrients, waste products, and maintaining the cell's internal environment. It's analogous to the skin of the cell, controlling what goes in and out. Antibiotics in this class work by binding to and disrupting the structural integrity of the bacterial cell membrane. This disruption leads to an increase in membrane permeability, causing the leakage of essential intracellular components such as ions, ADP, and nucleic acids. When these vital molecules leak out, the bacterial cell loses its ability to function, leading to its death. Compared to the other mechanisms we've discussed, directly targeting the cell membrane can be more challenging to achieve selective toxicity. This is because both bacterial and human cell membranes share some fundamental structural similarities. They are both composed of lipid bilayers. However, certain antibiotics exploit subtle differences in the composition of bacterial membranes. For example, some drugs might target specific lipids or charges present in bacterial membranes that are less common or absent in human membranes. The most prominent examples of antibiotics that work by this mechanism are the polymyxins, e.g., polymyxin B, colistin. Polymyxins are primarily effective against gram-negative bacteria. They are cationic, positively charged, cyclic peptides that interact strongly with the anionic, negatively charged, lipopolysaccharide, LPS, molecules present in the outer membrane of gram-negative bacteria and also with the phospholipids of the inner membrane. This interaction destabilizes the membrane, leading to increased permeability, leakage of cellular contents, and ultimately, cell death. Due to their potential for toxicity to human cells, e.g., kidney damage, polymyxins are often considered last-resort antibiotics, typically reserved for treating severe, multi-drug-resistant gram-negative infections when other, safer options are ineffective. Despite these limitations, they remain vital for tackling some of the most challenging bacterial pathogens. Their mechanism serves as a direct assault on the integrity of the bacterial cell itself. Now, we shift our focus to the critical challenge that shadows the remarkable success of antibiotics: antibiotic resistance. This isn't just a scientific concept, it's a major global health crisis, often referred to as one of the most pressing threats to human health in our time. So, what exactly is antibiotic resistance? In simple terms, it occurs when bacteria evolve and develop the ability to survive and multiply despite being exposed to an antibiotic that would normally kill them or inhibit their growth. Imagine an antibiotic as a weapon. Resistance means the bacteria have found a way to become immune to that weapon. This isn't a new phenomenon. Bacteria are incredibly adaptable organisms, and resistance is a natural evolutionary process. Whenever we use an antibiotic, we are essentially applying a strong selective pressure. Any bacteria within a population that happen to have a genetic mutation allowing them to survive the antibiotic will multiply, passing on that resistance trait to their offspring. It's evolution in fast forward. The problem, however, has been greatly accelerated by the misuse and overuse of antibiotics in both human medicine and agriculture. Every time antibiotics are used unnecessarily or incorrectly, we provide more opportunities for resistant strains to emerge and spread. The consequences of antibiotic resistance are severe and far-reaching. Infections become harder to treat. Common bacterial infections that were once easily managed can become life-threatening, requiring stronger, more toxic, and more expensive drugs. Longer hospital stays. Patients with resistant infections often need extended periods of hospitalization. Higher medical costs. Treatment of resistant infections is more complex and costly. Increased mortality. In some cases, infections caused by highly resistant bacteria can become untreatable, leading to increased rates of death. Threat to modern medicine. Procedures like surgery, chemotherapy, and organ transplantation, which rely on effective antibiotics to prevent post-operative infections, become much riskier. The golden age of antibiotic discovery has largely ended, and new drugs are not being developed fast enough to keep pace with rising resistance. This imbalance means we're rapidly running out of effective treatment options for some pathogens. Understanding how this happens is crucial for addressing the problem. So, given the selective pressure from antibiotics, how exactly do bacteria manage to fight back and become resistant? They are incredibly clever and adaptable organisms, and over millennia of competing with naturally produced antimicrobial compounds, they've developed a diverse arsenal of strategies to evade or neutralize antibiotics. These are the mechanisms of resistance. Think of it like a highly evolved defense system. When an antibiotic is introduced, bacteria that possess one or more of these mechanisms have a survival advantage, allowing them to thrive and pass on their resistance genes. We can broadly categorize these mechanisms into a few key strategies, each aimed at preventing the antibiotic from reaching its target or rendering it inactive once it gets there. The image on the slide illustrates some of these ingenious bacterial adaptations. You can see a bacterial cell that's developed ways to deal with the antibiotic, perhaps an enzyme breaking down the drug, a pump actively expelling it, or a change in the internal target so the drug can't bind effectively. These mechanisms are often encoded by genes that bacteria can acquire through various means, which we'll discuss next when we talk about the spread of resistance. But first, let's explore the individual strategies. We will focus on 4 primary ways bacteria become resistant. Enzymatic degradation, where bacteria produce enzymes that chemically destroy the antibiotic. Altered target site, where the antibiotic's intended target within the bacterium changes, preventing the drug from binding effectively. Efflux pumps, where bacteria actively pump the antibiotic out of the cell. Decreased permeability, where the bacterium modifies its outer layers to prevent the antibiotic from even entering the cell. Each of these mechanisms represents a different molecular strategy for bacterial survival in the face of antimicrobial challenge. Understanding them is key to developing new antibiotics and strategies to overcome resistance. Our first major mechanism of antibiotic resistance is enzymatic degradation, and it's a very straightforward and highly effective strategy employed by bacteria. In this scenario, bacteria produce enzymes that chemically modify or completely inactivate the antibiotic molecule. Think of it as the bacterial equivalent of a biological weapon defense system. They deploy a molecule that disarms the incoming threat before it can cause harm. The most famous and clinically significant example of this mechanism involves beta-lactamase enzymes. These enzymes are produced by many different species of bacteria and are the primary reason for resistance to the highly important beta-lactam antibiotics, which include penicillins and cephalosporins. Recall from our discussion on mechanisms of action that beta-lactam antibiotics work by binding to penicillin-binding proteins, PBPs, in the bacterial cell wall, thereby inhibiting cell wall synthesis. The core of these antibiotics is a unique 4-membered chemical structure called the beta-lactam ring. What beta-lactamase enzymes do is precisely what their name suggests. They hydrolyze, break open, the beta-lactam ring of the antibiotic. Once this ring is broken, the antibiotic molecule changes its shape and can no longer bind to the PBPs. It becomes completely inactive and useless. You can see in the image how the beta-lactamase enzyme acts directly on the penicillin molecule, breaking that critical ring structure. This renders the antibiotic ineffective, and the bacterial cell wall synthesis can proceed unimpeded, allowing the bacterium to survive and multiply. The prevalence of beta-lactamase enzymes is a major clinical challenge. For instance, many strains of E. coli and Klebsiella pneumoniae produce extended-spectrum beta-lactamases, ESBLs, that can inactivate a wide range of penicillins and cephalosporins, making infections caused by these strains much harder to treat. This mechanism highlights the molecular arms race between humans developing new drugs and bacteria evolving new ways to dismantle them. Our second significant mechanism of antibiotic resistance is the altered target site. This strategy involves a more subtle but equally effective bacterial adaptation. Instead of destroying the antibiotic, the bacterium changes the very structure of its internal component that the antibiotic is designed to bind to. Imagine an antibiotic as a specific key. If the bacterium changes the lock, its target site, the key no longer fits, and therefore, the antibiotic cannot exert its effect. This usually occurs through a genetic mutation that leads to a slight change in the shape or amino acid sequence of the target protein. This change is just enough to prevent the antibiotic from binding effectively, but not so much that the target protein loses its essential function for the bacterium. It's a delicate balance of survival and adaptation. A classic and highly relevant example of this mechanism is methicillin-resistant Staphylococcus aureus, MRSA. Staphylococcus aureus is a common bacterium that can cause a range of infections, from skin infections to more severe conditions like pneumonia and bloodstream infections. Methicillin and other beta-lactam antibiotics normally work by binding to specific penicillin-binding proteins, PBPs, in the bacterial cell wall to inhibit its synthesis. However, MRSA strains have acquired a gene called MICA. This gene encodes for a novel PBP, often referred to as PBP2A. This modified PBP has a significantly reduced affinity for methicillin and other beta-lactam antibiotics. Even when high concentrations of these antibiotics are present, they simply cannot bind effectively to PBP2A, and thus, cell wall synthesis proceeds normally. The bacterium remains resistant. This mechanism underscores the incredible adaptability of bacteria. By slightly tweaking a crucial cellular component, they can render an entire class of antibiotics ineffective, posing a significant challenge in clinical practice. Let's move to our third clever resistance mechanism, efflux pumps. This strategy is like a bacterial bouncer or a cellular bailing system, actively expelling the antibiotic before it can reach its target inside the cell. Efflux pumps are specialized, membrane-bound protein complexes found in the bacterial cell envelope. Their primary function, in a non-antibiotic context, is often to pump out metabolic waste products or toxins from the cell. However, bacteria have adapted these pumps to recognize and actively transport various antibiotic molecules from the cytoplasm back out into the extracellular environment. Think of it this way. The antibiotic gets inside the bacterial cell, but before it can bind to its ribosomal target, or inhibit DNA gyrase, or interfere with other internal processes, the efflux pump rapidly binds to it and pumps it right back out. This keeps the intracellular concentration of the antibiotic below the level required to be effective. These pumps are often multi-drug efflux pumps, meaning a single pump can expel a wide range of chemically diverse antibiotics. This is particularly problematic because it can lead to simultaneous resistance to multiple different classes of drugs, even if the bacteria haven't been exposed to all of them. Mechanism: efflux pumps typically utilize energy, e.g., from ADP hydrolysis or proton motive force, to move the antibiotic against its concentration gradient, from inside the cell to outside. Examples: efflux pumps are found in many important pathogens, including Pseudomonas aeruginosa, which is notoriously difficult to treat, and E. coli. They can confer resistance to tetracyclines, macrolides, fluoroquinolones, and even some beta-lactams. The constant pumping action means that even if a small amount of antibiotic enters the cell, it's quickly removed, preventing the drug from accumulating to a toxic concentration. This active expulsion mechanism is a highly effective way for bacteria to protect themselves from a wide array of antimicrobial threats, making it a significant contributor to multi-drug resistance. Our fourth primary mechanism of resistance is decreased permeability. This strategy focuses on preventing the antibiotic from even entering the bacterial cell in the first place, essentially acting as a fortified barrier. Bacteria can alter the structure of their outer membrane, in gram-negative bacteria or cell membrane, in both gram-positive and gram-negative bacteria, to reduce the influx of antibiotics. This is like a castle raising its drawbridge or sealing its gates to prevent an invading army from entering. For gram-negative bacteria, which have an outer membrane in addition to a cell membrane, this mechanism often involves changes in their porins. Porins are channel-forming proteins embedded in the outer membrane that allow small hydrophilic molecules, including many antibiotics, to diffuse into the periplasmic space, the area between the outer and inner membranes, and then into the cytoplasm. When bacteria reduce the number of porins, or alter their size and specificity, they effectively restrict the entry of antibiotics. This makes it harder for the drug to reach its intracellular target at a sufficient concentration to be effective. For example, some resistant strains of Pseudomonas aeruginosa or Enterobacteriaceae decrease or mutate their porins, leading to resistance to certain beta-lactams and fluoroquinolones. While this mechanism is more pronounced and better understood in gram-negative bacteria due to their outer membrane, gram-positive bacteria can also achieve reduced permeability by modifying their cell wall structure or membrane transporters to limit antibiotic uptake. This strategy is often combined with other resistance mechanisms, such as efflux pumps, to provide a multi-layered defense. An antibiotic might struggle to even get into the cell, and if a few molecules do manage to slip through, an efflux pump quickly expels them. This layered defense makes treating infections caused by such bacteria incredibly challenging. Now that we know how individual bacteria become resistant, the big question is, how do these resistance traits spread so fast and lead to the global superbug crisis? The answer lies in how bacteria share genetic information. They're incredibly adaptable and have two main ways to spread resistance. 1. Vertical gene transfer. This is simple inheritance. A resistant bacterium divides and passes its resistance genes to its offspring. So if just one resistant bacterium starts multiplying, you quickly get a whole population of resistant bugs. 2. Horizontal gene transfer, HGT. This is the real game changer. Bacteria can share resistance genes not just with their own kind, but even with completely different species. There are three main ways this happens. Transformation. Bacteria pick up loose DNA, like resistance genes, from their surroundings, often from dead bacteria. Transduction. Viruses that infect bacteria, called bacteriophages, can accidentally carry resistance genes from one bacterium to another. Conjugation. This is like bacterial mating. One bacterium builds a tiny bridge, a pilus, to another and transfers a plasmid carrying resistance genes. It's super efficient and can spread resistance across different bacterial types. The more we use antibiotics, especially when we overuse or misuse them, the more we help resistant bacteria survive and spread. The sensitive ones die off, but the tough ones stick around, multiply, and share their resistance genes. That's how superbugs emerge, and why they're so hard to stop. To wrap things up, we've seen just how powerful antibiotics are. They've saved millions of lives by targeting bacteria in clever and effective ways. But we've also looked at the dark side, antibiotic resistance. It's a serious, growing threat fueled by overuse, misuse, and bacteria's natural ability to evolve. Right now, we're in a race against time, and bacteria are catching up fast. So what can we do? Only use antibiotics when truly needed and exactly as prescribed. Don't push for them when you have something viral like a cold or the flu. Finish the full course even if you start feeling better. Stopping early helps resistant bacteria survive and spread. Never use antibiotics for viral infections. It doesn't help and only makes the problem worse. Support research into new antibiotics and alternative treatments. We need fresh tools in this fight. Practice good hygiene. Things like hand washing and safe food handling help prevent infections in the first place. Spread awareness. The more people understand this issue, the better choices we'll all make. Antibiotics aren't unlimited. Every time we misuse them, we make resistance worse. It's up to all of us to protect these life-saving drugs, so they keep working when we really need them. See you in the next lecture. Don't forget to subscribe my channel for more videos.